everyone. Today we are going to have a lecture on Kreb cycle, which is also called the TCN fully tricarboxylic acid cycle, and electron transport chain (ETC). Learning objectives for today is that by the end of this lecture, you should have explored what Kreb cycle and electron transport chain are. And we understand in details these two biochemical processes. And give an account, we should also be able to give an account of the ATP that is produced from tricarboxylic acid cycle and electron transport chain, or generally cellular respiration starting from glycolysis. Hopefully we have watched the lecture about glycolysis. And we should also be able to understand the role of oxygen in cellular respiration. To kickstart this, we start with the fate of pyruvate. Glycolysis form two molecules of three carbon each, and that's what we call pyruvate. And in other words, I mean pyruvate here is a 3-carbon molecule, so glycolysis would end up producing 2 pyruvate molecules in the process, and these molecules are further metabolized in the body, and it depends on the presence or absence of oxygen in your body cells. What happens in the absence of oxygen or limited oxygen in your body is that pyruvate will undergo anaerobic metabolism to form what you call the lactate or lactic acid. So pyruvate here is reduced to lactate by the NADH, and then catalyzed, and this reaction is catalyzed by pyruvate hydrogenase enzyme. Right down here is an illustration or a visual that you can refer to. However, there are very many sources of these visuals online. You can just type in the anaerobic respiration or conversion of pyruvate to lactate, and you have all the visuals you need. So second, during fermentation, pyruvate is converted to ethanol. That is a fermentation reaction that takes place. And lastly, in the presence of oxygen, which is going to be our major focus for today in TCA, is that pyruvate is oxidized by NAD and releases carbon dioxide and form acetyl, which is a 2-carbon molecule that combines with coenzyme A to form acetyl-CoA, and then it is channeled into the Kreb cycle or the TCA cycle. Right here is the visual also showing at the bottom of the screen right down. Have pyruvate been converted to acetyl-CoA. We are seeing the NAD coming in there and releasing NADH, and then the CoA there, the CoA, the carbon dioxide also being released from this, and CoA being combined or joined or fused together with acetyl to form the acetyl-CoA. So the summary of the fate of pyruvate is right here, that in anaerobic condition on the right hand in green here, on the left hand in green, it will form as a there's a mistake here, and acetyl-CoA is formed under aerobic conditions where there is oxygen. This is correct. Then lactate is formed in anaerobic conditions where there is limited or no oxygen in your cells, and then during fermentation you form ethanol. So this is basically the summary of everything that I've been talking about. A question can come and ask, describe the fate of pyruvate or explain the fate of pyruvate, and then you have to give us the 3 pathways that are fed during anaerobic condition, lactate, and all that. You necessarily don't have to describe the whole process of glycolysis, but you tell us what happens in the whole process of Kreb cycle, but you definitely have to explain to us how you arrive into forming acetyl-CoA. That would be the fate, and then you tell us acetyl-CoA will be channeled into the Kreb cycle. Now, to continue into the cycle point of focus today, we look at the Kreb cycle. This is a biochemical process that occurs in the mitochondrial matrix in the presence of oxygen. The acetyl-CoA molecule that has been formed, like we talked about earlier during the fate of pyruvate, that has been formed from oxidative decarboxylation of pyruvate, joined the Kreb cycle by combining with a 4-carbon molecule called oxaloacetate. And to begin this cycle, 2-carbon molecule called the acetyl-CoA combines with the oxaloacetate, which is a 4-carbon molecules. Then they form the citrate, that is a 6-carbon molecule. This is simple math here, that if you say 2 plus 3, it forms 6. So acetyl-CoA has 2 carbon, and then oxaloacetate has 4 carbon molecules. When you fuse the two or join the two together, they give you what you call the citrate. And the citric acid cycle then removes 1 carbon. This cycle will continue removing 1 carbon at a time in the form of carbon dioxide from this molecule until you form back the 4-carbon molecule, that is oxaloacetate, and that will mark the end of our cycle. And in that process, we shall be releasing in some NADH and a fat H 2 at some point, and also ATP, which we shall discuss as we continue during this lecture. So these chemical reactions here produces 2 molecules per glucose. So each of these Kreb cycle will produce 2 molecules per a glucose molecule. 1 glucose molecule will produce 2 ATP molecules. And you have 2 ATP molecules, and then the fat and the NAD, which we are going to discuss. Let's have a keen observation of that. The second step here, after the formation, after the fusion or the joining of oxaloacetate to acetyl-CoA, we form our citrate. So what happened next here is that this citrate here is converted into isocitrate. It's more like an isomer, and this reaction here is catalyzed by an enzyme called aconitase. What happened here is that citrate is isomerized to isocitrate. In simple terms, citrate is an isomer of isocitrate. So this process is a process that involves dehydration followed by hydration. And meaning in this first step here, you first dehydrate citrate, and then you form the cis aconitase. This is an intermediate between the two, and then the cis aconitase here is then hydrated to form the isocitrate. So in this case, you have citrate there, you remove water from it. That is what you call dehydration reaction, and then you form the cis aconitase. And after forming the cis aconitase, then we can now hydrate it and add back our water. And this reaction on both steps, so the hydration, the dehydration, are catalyzed by the same enzyme called aconitase. The OH group, which is the hydroxyl group in the citrate, is moved to a different carbon atom. That's where we get to understand what isomers are. Like we had an earlier lecture on carbohydrates, I think, and their structures. We briefly mentioned something about isomers. So they have the same molecular formula but different structures. This is exactly what you see. It is that, as you can see, that the citrate here, the citrate has, when you have a keen look at the hydroxyl group, and then the isocitrate also have a keen look at the hydroxyl group, you realize that they have happened in a way that they have been interchanged. The OH and the H on the citrate is interchanged, and I think just shifting it one up or the other and comes down. So that is basically what we call isomers, meaning citrate is an isomer to isocitrate. Then the third step in this reaction is oxidation of isocitrate and formation Like we said earlier on, in this reaction, it will continue by removal of carbon dioxide at the different steps, and until we form our oxaloacetate. And this is one of the reaction where we remove one of the carbon atom in form of carbon dioxide. And this step 3 here, the formation of the oxidation of isocitrate here, is a redox reaction, meaning it involves an oxidation and a reduction reaction, and decarboxylation here takes place in a way that carbon dioxide is removed uh, from the molecule. That's the intention of removing one carbon from it. And the reaction is catalyzed by isocitrate dehydrogenase as follows. First, isocitrate is oxidized to oxalosuccinate by NAD, and then it will release 2 hydrogen atoms, and this is where we're also going to form one of the NADH. And then, so one hydrogen, that is a proton, we shall call it proton. You will definitely find it in the electron transport chain. Let's just keep a keen follow-up of this. That we have one hydrogen atom, the proton, that is being transferred to the NAD plus to form the NADH. This NAD with plus is a reduced one, the NAD with H is the oxidized one. So in any way, if you hear me saying oxidized NAD, just what I refer to is the NADH. So it is transferred to NAD from electrons, and the remaining hydrogen or proton is released. So then third, here is the oxalosuccinate here that is being formed, is then decarboxylated to form alpha ketoglutarate. You can simply understand this in a way that our isocitrate is a 6-carbon molecule, and our alpha ketoglutarate is a 5-carbon molecule. So you can understand the essence or the relevance of decarboxylation here is that to remove one of the carbons so that we can form our alpha ketoglutarate. Right here is the summary of what I've been talking about, is that isocitrate that has been formed is converted or oxidized to form oxalosuccinate, which is then now converted or decarboxylated to form alpha ketoglutarate, a 5-carbon molecule in our reaction. Now the step 4 here is the formation of carbon dioxide and a alpha. This is supposed to be an alpha ketoglutarate oxidation. So it's a mistake that it's alpha ketoglutarate as per the heading there. But continuing with our lecture, we said the second redox reaction is catalyzed by alpha ketoglutarate dehydrogenase complex, and it involves a reduced NAD, a CoA, and an alpha ketoglutarate molecule. Now the product here, carbon dioxide and oxidized NAD succinyl CoA. It yields the second NAD and a carbon dioxide molecules. We write here, it's important for us to take note of these oxidized NADs that we are producing, because it's going to be very important in understanding our electron transport chain, which is still in this same lecture on. So let's get to look at this. Oxalosuccinate is then decarboxylated to form alpha ketoglutarate and yields carbon dioxide. I want us to look into this. We said this yields the second NAD. Where is the first NAD? The first NAD is being produced at this, the oxidation of isocitrate. So this is the second NADH that has been produced. So in this case, we have 2 NADH. And now we'll get to understand that pyruvate molecule, there are 2 pyruvate molecules that have been produced, and to go through it twice, so we'll get to multiply this by 2. However, we'll get to the accountability later. It's very important for our accountability and understanding of the electron transport chain. What I mean by accountability is the accountability of the ATP that has been produced, which is one of our objectives for this lecture. And the fifth step here is the formation of GTP or ATP and succinate. So GDP here is similar to ADP, and a free phosphate group reacts with succinyl-CoA. Succinyl-CoA synthase cleaves the thioester bond to release the CoA from its bond and form the succinate. And the energy release is used to combine GDP and inorganic phosphate to form GTP, which is similar to ATP. This is a source of energy, and what we say here is that we can now say one ATP molecule has been produced in this case, because the GDP or guanosine, G there is guanosine diphosphate, G is guanosine, D is di, then P is phosphate. Or this is guanosine diphosphate, and then a free phosphate group, and meaning this an enzyme called the succinyl-CoA synthetase here will cleave this bond of CoA with the sulfhydride bond. So this bond, the breakage of this bond here releases some energy that helps in the combining of GDP to inorganic phosphate to form our guanosine triphosphate, a molecule that is also similar to ATP. So this is very important to understand that there is an ATP molecule that has been produced here, and this is our first ATP molecules, and if I'm not mistaken, the only ATP molecules. However, we'll continue and do the accountability of it. This is the first ATP molecules that has been produced during Krebs cycle. And step 6 here talks about oxidation of succinate, and this is the third redox reaction catalyzed by succinate dehydrogenase. And succinate is dehydrogenated by fat to form the fumarate, which is a 4-carbon. So you just have to understand here that since we are going from a 5-carbon, we are going from succinate, which is still a 4-carbon, to fumarate. There is a reduction, there is a removal of hydrogen here, or the proton, electrons. So what happened is that this is fat today, which is in this reaction, it is fat that is our acceptor, the hydrogen acceptor for this matter, and this fat here takes up the hydrogen that has been produced from the redox reaction that involved conversion of succinate to fumarate, and we form our fat H2. We too have to take note of this fat H2, as it's going to be very important in our next part of this lecture. Then the seventh step here is the hydration of fumarate. Hydration is addition of water. This reaction is catalyzed by fumarase enzyme that helps in the hydrolysis of fumarates. That happens at the double bonds, as you can see in our fumarate. We have a double bond there, and I don't know if my cursor can come and show something right here on the left hand side where there's fumarate. It is stereospecific, so the only L isomer of this product malate is produced. So to hydrolyze in this, you only form the L malate on this product on addition of water. Then the 8th step and the final step is that we have regeneration of oxaloacetate. So this is a redox reaction as well. I think this will be the 4th. It is catalyzed by NAD and reacts with malate, picking up 2 hydrogen atoms, and should have said catalyzed by malate dehydrogenase. So this is the NAD plus here is reduced to NADH with the release of a proton and formation of oxaloacetate, which will now combine with another acetyl-CoA molecule to begin the cycle again. So once you have formed our oxaloacetate again, the cycle restarts and continues in the same way. All this step from step 2, from step 1, combining of oxoacetate with acetyl CoA starts over again. So right here is just some problem of error. It's a typo. It's a typo that we are seeing here. This redox reaction catalyzed by it's catalyzed by it's catalyzed by malate dehydrogenase, and you can get to understand here that NAD reacts with that and is reduced. It's reduced to. Let me go back to my presentation mode, and is reduced to NAD with the release of proton and the formation of oxaloacetate that can combine with another molecule of acetyl CoA to begin the cycle once again. So that is it. This is a redox reaction catalyzed by malate dehydrogenase. I think these statements now make sense. And then the reduced NAD here is reacts with malate picking up 2 protons and is reduced to NADH with release of hydrogen or the protons and formation of oxaloacetate that can combine with another molecule of acetyl CoA to begin the cycle again. So that's definitely the end of our description of this Kreb cycle. And now this here is a summary of everything of Kreb cycle, starting right here where the cursor is, brown coloring where I have acetyl CoA that is joining oxaloacetate, and the process continues. This is a summary, and as I said earlier, you can always get visuals from online. Your online sources are very many. They have very many things. They have a lot of these illustrations or images, visuals that can help you better understand the cycle. So understand that and master the cycle. In summary, we formed in NADH2, and then we formed FAD, meaning we have formed 2 FAD, two ATP molecules, and 4 NADH. So let's continue, and we'll get to do that. And then continuing to what called the electron transport chain. And this is the final step of cellular respiration, and it's one that results into the production of a lot of energy. Most of the energy, ATP, is yielded through this. And this reaction, the reaction in the citric acid cycle, also removes electrons which are passed to Sertoli molecules which are the NADH and the FADH2 for transport to the last stage of cellular respiration, the electron transport chain. This chain involves a series of molecules, most of which are protein associated with the inner membrane of mitochondria. So meaning these electrons are going to be passed in on from these electron carriers, which are the NADH and the FADH2. They'll be passed on and passed along all these protein molecules or this series of molecules, which we are going to talk about. We are going to see them briefly: the cytochrome, the Q cytochrome, and everything about it. So to continue in, these molecules accept the electrons from the Sertoli molecules, which are the NAD and the FADH, NADH and the FADH, and then pass them from one molecule to another down the chain until they are finally combined with oxygen to form water and carbon dioxide. One of our learning objectives was to understand the role of oxygen in this process. So as electrons are passed along, their energy is trapped and used to make ATP. So I guess we understand here that as this energy passes along this series of molecules, the energy is trapped here and is trapped and then used for the synthesis of ATP. in their body, which the body can now utilize. Now, key facts to note about the electron transport chain is that about 34 molecules are made from the product of one molecule of glucose, meaning one glucose molecule makes up to around 34 ATP molecules. Then the process is, sorry about that. The process is a stepwise movement of electrons from high energy to low energy that makes the proton gradient. We'll get to see the proton gradient coming in the process as we'll be describing the electron transport chain. And then the proton gradient powers ATP. Once we create that gradient, a proton gradient is like a difference in the concentration of these protons. The protons are the H plus, the hydrogen plus. And so the proton gradients for powers ATP production, not the flow of electrons. Meaning those electrons can flow, but that proton gradient only powers the production of ATP, but not the flow of electrons. Those electrons can flow without ATP. So this electron transport chain only occurs when oxygen is available, since we know it's the final electron acceptor. That's its role, and we'll get to understand it. So, electron transport chain here, the electron transport chain is the final and most important step of cellular respiration. And like I said earlier, it produces the most ATP molecules. And while glycolysis and citric acid make the necessary precursors, which is the NADH2, NADH and fat H2. And in here, the electron transport chain is where a majority of ATP is created, like I said earlier. So that's approximately 30 to 34 ATP molecules that have been made from this chain, or from this whole process of cellular respiration, from one glucose molecules. Now, electrons pass from complex to complex. Blue arrows, they power the movement of hydrogen atoms. Now, before I get into this, because you're seeing blue arrows and somewhere you're going to see pink arrows, let me take you right down here. Exactly, this is it. And this is an illustration of the whole process of electron transport chain generation. So whenever I say pink or blue arrows, I mean what this is. Blue arrows are the ones right here where you see complex 1, where you see Roman number 1, that's complex 1. These are the series of protein molecules that we have been talking about, where electrons are passed from one molecule to another until they reach oxygen and react with oxygen to form water. Then our ATP is formed. Proton gradients, we're seeing these electrons powers the pumping of the electrons into the intermembrane, and they creating the proton gradient. Once this proton gradient is created, at the end of everything, they sort to call, on the right hand side here where you're seeing ATP in a yellow or orange thing, I think this is just yellow, is that ATP is being generated from this, and this is what we call the ATPase enzyme. So the hydrogen or the protons pass through this and gets back, shuttled back into the matrix to balance this electron or the proton gradient. Sorry, the proton gradient actually, and then all that energy that they obtain from it. Sorry about that. All that energy obtained from it is used to shuttle all this. Is used to generate all this ATP. Now let's get to describe the whole of this process. You're looking at this, that we have the blue arrows and the NADH here are the donors, are the proton donors, the electron donors. They carry all those precursors needed for this reaction to take place. Now, at this reaction on the left hand side, you right see here that deep inside here is a matrix, and right up there is the intermembrane space, and this is a membrane, mitochondrial membrane. The mitochondrial membrane that is composed of series of protein molecules. These pink ones are where you're seeing 1, 2, and then also this blue is 1, 3, then 4. These are complexes. Then there is this yellow one with Q. This is called cytochrome Q. Then there is a green one here, is called cytochrome C. And this is basically the molecules where these electrons are passed from. When you look at the final thing here, right here is ATPase enzyme, a system, the part where the protons get back into the matrix and then ending up generating the energy. Now let's look at this process step by step. I may not go back into the notes right now. We first need to discuss on what is happening on this illustration before I get into the notes. I think that's most important at the moment. So let's pay keen attention at where my cursor will be moving, and I'll move it definitely slowly. Right here, or the cursor on the left hand side of this screen, where we have complex 1 with Roman number 1, a pink one, is having the nut that is nut now that has been oxidized into nut H. What happened here is that it releases this hydrogen. On releasing hydrogen, it gives away electrons to this complex 1. And the electrons are transported along this complex 1. That is what we call that is what you're seeing in the blue arrow, representing the electrons being carried. Electrons has been brought to this complex by NADH. So NADH here releases a proton and the electrons on it. So the electrons will be shuttled or carried along this complex and passed into the cytochrome Q. While this proton that has been produced or released from the NADH will be pumped from the matrix into the intermembrane space. This needs energy, but the energy from breaking off this higher proton and releasing of that electron there is powerful enough to pump that electron. So sorry, not electron, the proton. So the protons here passes or are shuttled up into the intermembrane space, and that's why we say it creates a gradient, proton gradient. Meaning, if all these hydrogen or the protons, hydrogen ions or the protons have been pumped into the intermembrane space, there is a concentration difference or gradient. There will be more of these hydrogen ions or the protons in the intermembrane space and less in the matrix. That's what creates the gradients or proton differences. Then sorry, my cursor seems to get lost sometimes. Okay. Then second, these electrons being carried from cytochrome from complex 1 to cytochrome Q. FAD also brings its hydrogen and electrons and donates it into cytochrome 2. We need to get into this difference and understand that FAD H2 brings its electrons at a much lower energy levels, where on complex 2, and NADH brings it at complex 1. You understand that at the end of it all, there is a difference in production. This one produces like about this FAD, each of these FAD H2 here will end up producing 1.5 electrons, ATP molecules, and then the NADH will end up producing like about 2.5 ATP molecules each. So we'll be able to do that calculation later. But bottom line here is that all these two electron donors, the FAT H2 and the NADH, brings in their electrons and then pass them onto these complexes, and the NAD passing it to complex 1 and FAT passing it to complex 2. On this process, the electrons are passed also and moved along. So both of them here passes the electrons to cytochrome Q. So these electrons are at cytochrome Q continues and are passed on to complex 3. And also more of these protons are pumped back into the intermembrane space. You will appreciate this later when we get to account for when we get to know how ATP or ATP is being produced here on this same process. So these electrons from complex 3 will be passed on and shuttled into cytochrome C, and cytochrome C will shuttle it back into the intermembrane, into the matrix, where now these electrons meet oxygen and they form water. Then now what happens to this hydrogen, which is the protons, the hydrogen ions or the protons that are in the intermembrane space, is that they are shuttled back, they are shuttled into the mitochondrial matrix, and this occurs with the help of ATPase enzyme. So as these hydrogen ions or protons are shuttled back, the ATPase enzyme helps synthesize ATP molecules from here, and that's how we end up producing energy from electron transport chain. Now let's get back to our notes that electrons pass from complex to complex, which is complex 1, 2, and 3, and also our cytochrome Q and C, and they power the movement of hydrogen atoms, which are the pink arrows, we talked about them, and into the intermembrane space. What happens that electrons, as they move, they power the movement of hydrogen atoms. uh Let me look here. You see this pink arrow, hydrogen atoms, hydrogen ions, they are pumped up, pumped up. So that is what happens here. Then NAD, H, and fat H2 are the non-electron carriers. They carry the electrons. This means they are capable of donating electrons to the transport chain. We looked at it earlier in our process of Kreb cycle that we formed NAD, H, and fat H2, and they carried our electrons and the hydrogen that we have been removing from all those intermediates that we are able to form these compounds. So what happens that it means they are capable of donating electrons. So what happened here is that they donate electrons. Let's look at this. NAD H donating to complex 1 and fat H2 donating to complex 2. And now the protons are shuttled back into the matrix and allows ATP synthesis to make ATP, which is actually our molecule, the last molecule here. This is ATP synthase. It synthesizes ATP by addition of inorganic phosphate to ADP. So that is basically what we can say about the process of electron transport chain. Hopefully you have understood and you can define, or you can describe, sorry, you don't need to define the process, you can describe electron transport chain. And now let's look into the role of oxygen. What does oxygen do? When we follow the path of electrons, at the end of the chain the electrons are taken up by oxygen molecules to make water. This is why oxygen is known as the final electron transport chain. So oxygen being the final electron acceptor, just to put things into perspective, when we breathe in oxygen with our lungs, then it is they transport it with the red blood cells into our arteries and to the cells, then it is ultimately used inside the mitochondria of every cell to accept electrons at the end of the electron transport chain. Meaning in every cell the electron transport chain is occurring. So as you breathe in right now, the oxygen is taken, you take in your oxygen, it dissolves in the lungs and get absorbed, transported by the red blood cells and then into the site for use. And then now electron transport chain is happening in the cells that is going to produce all this energy. What happens? So that's it for that, and we need to account for where do the 30 to 34 ATP come from. There are two net ATP. Two net ATP are made in glycolysis. Like also we have to agree to that we have to agree the fact that there are four ATP molecules produced during glycolysis, but then two have been used during the process or at the beginning. So you will definitely have one person who will definitely have from two ATP. So we'll have two ATP molecules as the net, that is 4 minus 2. Now, based on a lot of experimental work, it appears that four hydrogen ions must flow back into the matrix through ATP synthesis to power the synthesis of one ATP molecules. So this is where the energy production during electron transport chain comes in. That all the hydrogen ions will produce one ATP, four of them. And sorry, I didn't talk about the Krebs cycle. Two are produced also. Two ATP molecules are produced from the Krebs cycle, meaning we have four ATP molecules at store. Two from glycolysis and two from Krebs cycle. Then now in the electron transport chain, the energy depends on the number of NADH2 or NADH and fat H2. Now every molecule of NADH2, every molecule produces 2.5 ATP, and every fad H2 molecule produces 1.5 ATP molecules. Now electrons from fad which enter the chain derive pumping of only 6 hydrogen, leading to production of what? 1.5 ATP. Now let's look at this. We saw the NADs, we saw the fads, where they come from and how many they are. So each NAD yields about 2.5. We will get to know, if you've forgotten how many, we are going to come there and you know how many fad, how many NADs are there. So with this information, we can do a little inventory or calculation or mathematics for the breakdown of one molecule of glucose. One molecule of glucose molecule, what does it give us? So let's get into it. Let me give you a summary of everything. That is cellular respiration here. Right up there is that we are seeing glycolysis, and glycolysis being connected now here to the link reaction. A link reaction, that's where pyruvate is converted to acetyl-CoA, and then on that, after converting it to acetyl-CoA, we have our citric acid cycle, the Krebs cycle, and then finally we go to electron transport chain. Looking at this orange, can I say pinky, purple, all these colored arrows here, is that you're seeing two NAD H, and then you're seeing 6 NAD H and 2 fat H2. So this is exactly what happens. Let's look at this. That in glycolysis we have 2 ATP molecules, and that is 2, and 2 NAD molecules, which will give us a rate of 5 ATP molecules. Then pyruvate oxidation here is that we have 2 NADs and 5 ATP, because we say 2 NADs will produce 5 ATP. That is 2 times 2.5. If we said 1 NAD molecule produce 2.5, meaning 2.5 times 2 will give us that. And then this also will produce 5. These are 2 ATP molecules, and then 2 ATP molecules that were readily produced during glycolysis. And when you come to Krebs cycle here, we have 2 ATP molecules, 2 ATP molecules that will produce what? 5 citric acid molecule. 2 ATP molecules, which is the GTP that we talked earlier about, one of our description earlier. And then we have the 6 NAD molecules, that is 6 times 2.5, that will give us 15. And then we have the 2 fat 2, that is from citric acid, that's like 2 times 1.5, that's why we have the 3 ATP molecules. So this all in all will result into our numbers of ATP molecules. If you consider that, I think it's 32 or 34 ATP molecules, and if you look at this side strictly, it's around 30. So I want to say thank you very much. This is a summary of the cellular respiration, and thank you very much for today. And right, you can see up there is the glucose being channeled into glycolysis, and then you have pyruvate into fermentation. That is, then in absence of oxygen, we have lactic acid. Then in presence of oxygen, you go now into mitochondria. Right here, this orange thing is the structure of the mitochondria. And then acetyl-CoA is from there, TCA from there, electron transport chain is still in the mitochondria, and then you release your ATP, carbon dioxide, water. All this carbon dioxide is what we exhale out from metabolism. Questions, friends? Describe the process of TCA and ETC. I hope you now understand those abbreviations. Explain the role. TCA is tricarboxylic acid cycle. ETC is electron transport chain. Explain the role of oxygen in cellular respiration. Explain why fat produces less ATP than not H2. You have to explain that, and then account for the ATP produced during the process of cellular respiration, which we already accounted for. And with these few slides or the lecture that we have, I want to say thank you very much for being patient and attending this lecture up to its end. So thank you very much, and this is where I can stop. Thank you.